Today we're going to look at a course overview. Our objectives are to make the transition from molecular events to the organ systems, to have classical histology merge with modern cell biology, and to learn new techniques, types of vision, both for light and electron microscopy. We want to learn the relationship between the structure of cells, organ systems, and their functions. And then we want to learn normal histology as a prerequisite for pathology, and then learn how to communicate and have fun with histology. Thank you. Course overview. Biology of mammalian cells and tissue. Hi, I'm Larry Johnson. I am a professor at Texas A&M University. And today we're going to talk about an overview of this histology course, which is an undergraduate and graduate school histology course. We'll start with cells, and then we'll continue with tissues, and then we'll talk about organs and organ systems. Now, here is an orphan website where there is a PowerPoint presentation to all these presentations, as well as the clinical correlations. There are some electron micrographs and some question guides, as well as items to identify for each of these presentations, where it's muscle, nerve, connective tissue, or the different ones. So this is the biology of mammalian cells and tissues, 443 and 602 for undergraduate and graduate students. We have a host of people that participate. We have some guest lecturers in this course, and this manual we'll be using, it came from Southwestern Medical School whenever I was a faculty member there. So as I said, I'm a professor in veterinary integrated biosciences, and we have other instructors that help out with this course as well as guest lecturers. So if we look at today's date, you'll see an overview of the course and also introduction to the writing, because this is a W course. For the undergraduate students, it's a writing intensive course. You get a credit for writing intensive in this course. As you can see, we have lectures and we have labs, and then we have a series of conferences, and then we have clinical correlations is another activity that we have. And here you can see where the writing components are required, certain dates, and then also when we have quizzes. We'll be having a quiz next Friday, Friday week that is, and so be ready for those, and those correspond to the page numbers. Now, the objective to this course is to make the transition from molecular events to organ systems. And here we see a cut through the human head. We can see the brain, the skull, skin, eyes, nasal cavity that we can see there. We want to have classical histology merge with modern cell biology, and to learn techniques and new types of vision using light and electron microscopy. We want to learn the relationship between structure and function, and have the organ architecture facilitates its function. We don't want to look at normal histology as a prerequisite for understanding pathology. You're probably too young to have pathology, but you might know some of your relatives that have had problems before you, to help you to understand pathologic conditions, and also to learn how to communicate and have fun with histology. We have a laboratory notebook and files. We have study guides and items to identify. Those were on those sheets I mentioned to you. We also have a course schedule, tell you what we're going to have all the times. We have computers to review some of the images, electron micrographs, and PowerPoints for the introduction to labs, conferences, and lectures we have for you. And so here you can see where we start out with cells in the lab. We look at cell structure 1, cell structure 2, cell structure 3 to go through the organelles. Then we start with the nucleus, and then epithelium, blood, connective tissue, the various components, as you can see. And these are writing assignments and the quiz assignments. And we will have introduction to lab next time, as we can see. So we have clinical correlations. As you can see, spring break was to cover hematopoiesis blood and lymph vessels, the endocrine system, lymphatic system. We'll also have lab practicals and didactic practicals and the whole gamut, male, female reproductive system, as well as the eye, digestive tract, respiratory tract, integument, the different things that we're going to cover. This is a course evaluation for each of these items. For the lab, conference, or clinical correlations, you'll have a chance to score these ineffective to very effective. Also, we have a list of the slides that we have and URLs that go to each of these slides. And here we see there's we have electron micrographs. This is one right here showing the Sertoli cells and germ cells that we see there. These are primary spermatocytes, spermagona, and spermatids that we can see. We also have electron micrographs of the whole sort, so you can be able to see. We also have on a YouTube channel, which is VIBS Histology YouTube, videos that go for the introduction to the labs, and also right there are access to the PowerPoint slides that you have there. So if you look at the playlist, you can see this is the basic one that we have. This is one for med school, but this is the one that we have for the undergraduate and graduate histology. This is for the freshman undergraduate program. We have the conferences. We have the basic for you to get the basics, and you can see the access to the PowerPoint slides and the images thereof at that site, which will be here. It will show you the different labs, the conferences that we have, information about that. All these are things that matches the they match the YouTube channel, whether the med school, the freshman class, or the senior and graduate class that we can see. So introduction to microscopy, use your atlas because things are already labeled for you. You can identify the cells. We have outlines, laboratory exercises, word slides, guides for you. This is all an evaluation must be turned in. There are textbooks that are recommended for you to get here. Earlier versions of the book is probably all right. We'll have 26 lectures beginning at a certain time. And then we have laboratory that's associated with those lectures, and we have 10 conferences to develop conceptual thinking with problem solving is the purpose of those. We have preparative homework to allow a student to become familiar with the subject prior to that time, and preparative homework, about 25 sets I'm due at the time of the lecture on. The homework consists of 3 to 5 structures or structural features of whatever we're covering that particular day, and how those features facilitate the overall function of that. Like, for example, a structure could be the lens of the eye, and the function of the lens of the eye is to focus the image on the photoreceptor cells in the retina. And so that contributes to the overall function of the eye, which is to observe the visual world. So that's what you need to do. And then we're going to have the course flipped, basically. You will watch a short video presentation and watch the PowerPoint slides that you can see right here, here, and here, and then come to class and you'll be set for activity. We'll have introduction to conferences in that flipped format. Here you can see where the Navy has a flipped laboratory. And we have tests for these materials, practical exams. We have essays, and the didactic would be multiple choice. There'll be a term paper associated with a writing assignment here. To advance understanding of conferences are needed to improve the communication skills of students and to improve the awareness of issues related to common diseases. That's what we're looking for. So you choose one of the topics that we have for our conferences and write something for your professional colleague, your colleagues, and it has to be at least 2,000 words associated with that. Paper references have to be cited as if a journal would use that. And then we have a writing assignments, conference essays as part of your writing assignment that you have. And so basically the writing assignments and all this is an outline, description, first draft, second draft, final draft. All these are different times, and the times have been designated for you in the schedule. And these are the points associated with each of these. The main thing is the term paper itself, and 50 points. A W course has to have 250 points associated with, or 25% of the grade has to be a writing assignment, and you must pass the writing assignment before you can pass the course. Exam, we have quizzes every other week, practical exams, concept identification, essays for conferences, and didactic to test everything. Here we see how the grading goes along. Preparative homework is about 50 points. 6 quizzes, you can drop into 2, so it's 200 points, 50 points each. Writing assignment is 200 midterm. As we can see there, the conferences is another 50. And so for midterm and final, we have a practical identifying things and what does it do. Conferences, essay, and didactic and multiple choice. We'll repeat that for the midterm and the final. Student participation in class and student enhancement of the objective of the course will be appreciated and may pay off in borderline cases. Scholarly dishonesty is not allowed, and grades may be curbed if they need be. Getting started with microscopy. One of the most important things about microscopy is color illumination, and there's basically a few steps associated with color illumination. Basically, the first step is you focus, you put the specimen in there and focus on the tissue. And then you close the field diaphragm. And then you focus the condenser. Here's the knobs here. And then you center the condenser, and then you open the field diaphragm. If you close it, you got to open it at the end, and that will be better illumination, color illumination, that will be. And then still we can see a piece of tissue here. And so we can come right here and click on that 220 right there, and we will see the piece of tissue. This is a little boy's finger. You can see the cartilage and the bone, a bone here, cartilage, and the skin around it. And here it is. And you can see how you can manipulate it. You can look at the cells inside. These are bone marrow cells. These are bone marrow cells. These are bone cells, and this blue here is cartilage, as we can see. So you can change the magnification of this if you want to. You can look around for the different kinds of cells that may be there. There's some fibroblasts over here. These are chondroblasts inside of the cells here, chondrocytes inside of the cartilage, and then osteocytes here, osteoblasts along the edge that we could see in here. And then we see some other cells, multinucleated cell right here, osteoclast. This is the one that eats down the bone. So you can see that. If you look here at the skin, you can see the epidermis and the dermis. There's the blood vessels of the dermis below. These are the sweat gland ducts going to the sweat glands that we see there. This would be the little boy's fingerprints in through there. And there's, of course, bone in your finger. That's where your fingernail would be. And here is a tendon that is running from that digit that's going to ultimately run to a muscle. So you can, and there's also tendons on this side as well. So that's it. There's another one showing skin. There's another one showing scalp, and you can see that this guy had curly hair because you can see that the hair follicles themselves are curly. Here we see a little piece of tongue. We can see the epithelium on the surface. We can see the muscle. And if we look right here, we can actually see glands. So this is part of our fluid that's inside our mouth, the saliva. These glands here are producing those. And then these are muscle cells. And you can see they're long cells, a nuclei on the periphery of them, and you can see the capillaries that are running down through there. Very nice. Capillaries branching and running down through there, and we can see those as well. And then this is skin. If you pull the epidermis away, you will see the dermis. And so we can click right here, and we can see the dark. So if you were where the dermospore is looking out of cells, you'd be looking through the epidermis, and this is the epidermis, and through here. And what you see is cells are stained for enzymes associated with making melanin, and you can see how these are dendritic cells. These are melanocytes. There's this nucleus there that passes the granules onto the keratinocytes. These would be hair follicles or something in through there. And so we can see in Port Aransas, there's a whooping crane that we see out through there. Birds of all kinds. This is Rockport, some birds early in the morning. This was early in the morning. I got up. My wife was still sleeping, but I got up to take that picture. Birds on the wing, and there's a whooping cranes. Yeah, it's a very nice one. The one is more reddish, as we see here, is actually a juvenile one. Here we can see it very nicely. The reddish here, that's a juvenile whooping crane, and there's some ducks, as you see. And there's evolution of science. As science continues, there's a lot of names in science which are men, but now women are taking over science as they should. We got lots of women into science because we need both genders into science. I hope this is useful. Take care. Bye-bye.